Welcome, everyone, and thank you for joining us for today's session on pioneering our understanding of the human brain. I'd like to introduce Dr. Michael Beauchamp and Dr. John McNaughty, who will be co-presenting their talk, RAVE, Our Visualization of Intracranial Encephalography. Dr. Beauchamp is the professor and vice chair of basic research in the Departments of Neurosurgery and Neuroscience and the director of the Core for Advanced MRI at Baylor College of Medicine. Dr. John McNaughty is an assistant professor of neurosurgery at Baylor College of Medicine. For a complete biography on our speaker, please visit the Biography tab located at the top of your screen. I'm Dr. Roshana Kashimir, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. We would like to extend our special thanks to the Brain Initiative program directors, and in particular, Dr. James Natt, for their efforts in organizing today's session. Before we begin, I would like to remind everyone that this event is interactive. We would encourage you to participate in submitting questions during the presentation. Simply click on the Ask a Question box located at the far left of your screen and type your questions in the drop-down box. Our speaker will respond to your questions by email following the presentation. This presentation is educational and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Beauchamp and Dr. McNaughty.
Thanks very much for joining us today. My name is Michael Beauchamp, and I'm going to be telling you about a software package that we're developing known as RAVE for our analysis and visualization of intracranial encephalography. The development team for this package consists of myself and my colleagues John McNaughty and Zhengjia Wang, and the funding for the development of RAVE was provided by the NIH Brain Initiative. So the outline of the talk today will consist of two parts, and the first part is a PowerPoint presentation which will take you through some of the background and development goals of RAVE, and the main part of the talk will consist of a demo of the software package itself presented by my colleague John McNaughty. So the first question to answer is, what is RAVE useful for? And essentially it's useful for analyzing brain data recorded from electrodes implanted in the brains of human participants. This technique has a number of different names, intracranial EEG, also ECOG when we were referring to grids or strips of electrodes, or stereo EEG, which is the name used when we insert penetrating or depth electrodes into the brain. And the RAVE logo contains both kinds of electrodes. And the R and the E, those are strips of electrodes, and the A and the V, those are meant to portray depth electrodes. And as you can see in the graph, this technique is growing rapidly in popularity with many hundreds of new publications every year devoted to this method because it has a unique combination of high spatial and high temporal resolution, which is important for understanding the brain. Why we need RAVE is best understood with analogy to another new technique, fMRI, which started to become popular in the mid-1990s. At that time, there was no software packages for analyzing fMRI data, so each lab was forced to develop their own set of idiosyncratic tools. This was a big obstacle to progress because this was difficult and scientists had to spend a lot of time developing software tools, which was not their area of expertise, rather than doing experiments. So with the creation of purpose-built fMRI software like FSL, Brain Voyager, and especially AFNI, which is developed by one of our collaborators, Bob Cox, non-programmers and non-engineers could do experiments and analyze fMRI data to make discoveries about the brain. And we're at a similar point in intracranial EEG right now. Most labs are using a hodgepodge of software tools that they've downloaded from the internet or MATLAB code that they've written themselves. And this presents a serious obstacle to progress because grad students or postdocs or PIs have to spend time writing MATLAB code, which maybe they're not well equipped to do, or develop statistical tests instead of analyzing data and making discoveries about the brain. So our goal is to create a purpose-built software package that's designed solely for intracranial EEG and has the flexibility necessary to make discoveries about the brain. And our development goals for RAVE, our most important goal is to keep users close to the data. Because the brain is so complicated and there's so much we don't know about it, it's absolutely critical that you have the ability to examine raw data and make sure that you're not misled by artifacts. There can be artifacts in intracranial EEG data like in any other kind of data, and if you're only looking at the final product of the analysis, which a long analysis pipeline might spit out, you might not realize that somewhere along the analysis pipeline, one of the steps had gone awry, and the final map that you're looking at is absolutely meaningless. So the good thing is that computers are so rapid now that we can do real-time interactive data analysis without leaving it to run overnight, which you might have had to do 20 years ago with fMRI data. So you can easily update your analysis in real time and see what happens. And this makes it easy to analyze. If you have the right software tool, it makes it much more efficient to analyze data. Second, we want to have rigorous statistical methodology. Again, by analogy with fMRI, there's been periodic paroxysms of controversy in fMRI. For instance, in the early days, subjects were treated as fixed effects, when it eventually became clear that really no, subjects were not all identical. They needed to be treated as random effects, and many other kinds of statistical arguments. So the part of this comes because people are making arbitrary choices who have not necessarily had any training in statistics and are just using a toolbox or a MATLAB function. So by building a purpose-built program, we can make wise choices and encourage people to do analyses the right way. Of course, we're not prescriptive, but we can say, okay, the built-in way that RAVE analyzes data has been vetted by statisticians and decided that it's a rigorous way to analyze data. And the final goal of RAVE development is to make sure that it plays well with others. That is to say that it allows you to work with existing tools that you might have in MATLAB or C or Python and integrate it into your existing processing pipeline so that you don't have to give up all the tools that you already know and love. So the current state of RAVE development is that we have more than 10 statistical modules, over 30,000 lines of code in the code base, and it's being used on a daily basis by many labs at Baylor College of Medicine. And of course, we would love for other users to use it as well, and you can download it for free from GitHub or from the website that's shown on this slide. And we're working on RAVE every day to try to improve it. So in the second part of the talk, you're going to hear my colleague going through some of the awesome features of RAVE, and we're going to demonstrate it on some real intracranial EEG data that we collected and that was published in a paper in eLife in 2018. And I'll just briefly introduce that data set to you to make it easier to understand when you see it analyzed within RAVE. So this is a study where we presented patients with 3 kinds of stimuli. These were short video clips of words. So as you can see in the left part of the screen, it could be a video clip where you could both see the face of the talker and hear the talker. So for instance, the talker would say the word drive, and you would see their, in this case, my former student Audrey Nath, you would see her lips move. In the middle condition, the visual-only condition, you wouldn't hear any sound. You would just see the face of the talker move. And in the final condition, auditory only, you would not see anything except a white fixation point on screen, but you would hear the word drive. So those are the 3 main conditions. And we went to St. Luke's Episcopal Hospital, where we worked with 8 different patients who had had electrodes implanted in their brains for the treatment of medically intractable epilepsy. After they recovered from the surgery, they rest comfortably in their hospital room. And if they wish, they can participate in our experiments, and that involves them looking at a screen, as you can see on this cartoon here, and holding a mouse to make behavioral responses. So in this experiment, they would see 30 to 60 repetitions of each of the words in each different condition, and they would press a mouse button when they heard a specific word, in this case the word press. So these patients had electrodes implanted in their brains. As you can see, we created a cortical surface model of the brain. Each red dot shows one electrode. In the bottom left of the slide, you can see the pictures of the grids or strips before they were implanted in the brain. Each of the grids or strips has wires that are channelized out of the patient's head. So you can see the wire coming from the top of the bandage in the middle cartoon and fed into a BlackRock system where the data is recorded to a computer hard disk. After we collect the data, we can feed it into RAVE and do many different kinds of analysis. Just to give you an idea of the kind of analysis, 2 kinds of analysis. That raw voltage signal, the red trace, shows the data from one electrode over the course of a second or 2. The y-axis on that plot is the voltage recorded from that electrode, and the little single frames of the movie represent the onset of a movie. So we could either do a voltage analysis and calculate an event-related potential, a sort of classic EEG analysis, as shown in the bottom left. Or we could do a time frequency analysis where we do a spectral decomposition of that voltage signal. And as you can see, the x-axis or time is the same, but the y-axis now has been transformed into frequency. And you can see there's a big blotch of red color showing that there's a large amplitude increase in these high frequency broadband activity from around 40 or 50 hertz all the way up to 150 or 200 hertz when the movie begins. So I want to emphasize that one of the big challenges of intracranial EEG data is we have so much data. There's many participants, hundreds of electrodes. We're sampling at up to 30 or 40 kilohertz in some cases. We have many trials, many conditions. So we need special software tools to analyze this data. And the tool that we're going to demonstrate to you, again, is called RAVE. And I will now pause the presentation and turn it over to my colleague, John Magnotti.
All right, thank you, Mike, for introducing the software package. As you can see on the screen, this is the main loading page for RAVE. RAVE consists of a set of modules that allow you to both preprocess, visualize, and analyze intracranial EEG data. Just to orient you to the screen, on the left side is the modules pane. Currently, we're looking inside the Condition Explorer set of modules at the Power Explorer module. And there are different modules in RAVE, as Mike alluded to. You can look at voltage, you can look at phase, you can conduct group analyses across subjects, and there's also some metadata that you can look at. But let's jump right into the Power Explorer. So the top left shows the electrode that we're currently looking at. It's electrode 22. We'll orient you in a minute to where this electrode is. And you can see just to the right of the electrodes panel, this is a spectrogram just like you saw in the last PowerPoint slide. So what RAVE allows you to do is interactively surf through spectrograms very easily. So in the middle left pane, I'll scroll down a little bit so you can see it, we can select both the frequencies of interest, the baseline window. This is a period of time when the subject is doing nothing relative to the task. And then also our analysis window. This is the region when the video is being presented. So that's how we get this black box that's drawn on top of the spectrogram. One of the interesting things about ECOG relative to other techniques is that on each individual trial, we can actually see significant brain activity. And so here in the middle panel, we're plotting this average power inside the frequency range of interest on each trial. So the y-axis here, each row is an individual trial. I've labeled the trials by what video the person is watching. These particular videos have a blank screen, and they're only hearing the word that you see. So at the top is last, then meant, known, and then drive. And what you can see in these plots is that there's a very consistent increase in what we'll call high gamma power a little bit after the auditory comes on. For all these plots, time 0 is the start of the acoustic sound. And it's interesting that you can see that there's variation across the stimuli, but by and large there is great similarity in response to each stimulus. As we continue to scroll down, RAVE shows a time series plot. Shows you that during the baseline period, this is controlled to be 0, or to be mean 0. And then right after the auditory onset at time 0, the power increases, and then after about a quarter of a second starts to decrease. And this analysis window defines the time at which we're going to do our statistical comparisons. So the final plot on the bottom right, this shows you for each individual trial, this is the average power within that red window. So each point here is the trial response to a particular stimulus. And by default, for a single condition, we can just compare that activity to 0 to see if this electrode had a significant response to these auditory-only stimuli. Now, of course, we can go back up to the top and change the electrode we're looking at. For example, what if we look at an electrode that's in the auditory cortex, or excuse me, the visual cortex? So here, electrode 78, that's in the visual cortex, we can see that there's not really a strong response at all to the auditory signal. very slightly elevated above 0, but compared to the previous channel, there's really not anything going on. However, what if we look at the visual responses, the visual stimuli? So I can go over to the trial selector and start to add in these other trial types. I'll click all of these. I can apply a label, call these visual. And what you can immediately see is that when we add in a second group of trials, that RAVE now shows these side by side. And so although there isn't a very strong response to the auditory stimuli, there's a very strong response to the visual only stimuli. So these are the stimuli where you just see the person talking, but you don't hear anything. And when we go back down and look at the time series, now we see both time series overlain. And then in the bottom right graph, we can see that here there's a much stronger response to the visual trials compared to the auditory only trials. And so we could do this for all the electrodes that we've loaded, but RAVE also has the ability to take this contrast that we've defined, this auditory versus visual contrast, and apply it to every electrode that has been loaded on the brain. So this takes a few seconds, but I've already preloaded this calculation. And so here you can see really nice the topographic organization of this contrast. So towards the bottom right in the superior temporal gyrus, there's a lot of red. So these are regions of the brain that respond more to the auditory stimulus than to the visual only stimulus. As we rotate the brain and look at the back of the brain, we can see that there's a lot more blue. And so these electrodes are responding more to the visual stimulus than to the auditory stimulus. Now using RAVE, I can click on these different blue channels. So this blue one here in the middle is electrode 78. And that allows us to then scroll up and see, okay, yes, that's the electrode that we're looking at now, electrode 78. But we can find other electrodes that we want to inspect. So electrode 22 is the electrode we were looking at initially. But let's look over at, for instance, electrode 23. So we can simply type in 23 into the electrode screen, and RAVE automatically updates to show you the new calculations for this new electrode. And what's interesting, if we scroll down and look at the time series, we can see that although the response to auditory only is much higher than the response to visual only, there does seem to be a response to visual only trials. And so what you might want to do is, what if we delete the trials for auditory only? Now RAVE will update to only show the visual only trials. And if we look at the comparison here, although the response is smaller, it's still significant. So in the bottom right panel, we can see again the individual trial plots, and the mean of this is 63%. So there is a significant change, significant response to visual stimuli, even though auditory is much stronger. And this accords with what we know about this region of the posterior superior temporal gyrus as being multisensory. And so it's responding both to auditory alone and visual alone stimuli. And so you could repeat this process with all of your different subjects. We don't have time to show you today how RAVE is organized, but there is a select data button that allows you to quickly choose which subjects that you want to load. And this is organized by projects. And so I'll just show you in the drop-down here, based on which project you want to load, we're looking now at the congruency data set, but you can see we have lots and lots of data sets that we've analyzed with RAVE. And even within this congruency data set, we've already analyzed lots of different subjects. So we are using RAVE on a day-to-day basis. The last step that you might want to do in your analysis is to try to look at some of these comparisons across all of your subjects. So I'll jump over here to the group analysis module on the far left and choose the linear mixed effects model. So we've loaded up in our mixed effects model module all of our subjects from the congruency data set. And what RAVE allows you to do here in this model building panel on the middle left is actually choose how you want to model your data. And so we'll model our data with the dependent variable being power, look at a fixed effects of conditions. So it's going to compare auditory versus audiovisual, and it's also going to include some other trial types that we had in this experiment. Here it's showing you in the formula panel the exact formula that's being used. And so this way, if you need to report in a paper, how are my statistics actually done, you can just you can say this is exactly the command that was run. We choose run analysis, and depending on how much data you have, it may take faster or slower. And we can see here that the LME output panel, RAVE now reports everything you need to know to compare condition across all of your subjects. And it appropriately takes into account both the fact that subjects are a random effect, but also that electrodes are nested within subjects. And so this is part of our goal to present more modern statistical techniques as just the way things are, just the way RAVE works. So we've shown you today, just to reiterate, that in RAVE we have a modular interface that allows you to compare conditions using power, voltage, phase, and then once you've made those analyses, you can export them to the group level and then see if the differences that you see at the individual electrodes are consistent across patients. Thank you.
Thank you, gentlemen, for that presentation of this cutting-edge work. As a reminder, our speaker will follow up with any questions about their presentation via email. Coming up next, Dr. Beauchamp. He will present Human Brain Science, the intersection of translational neurotechnology and systems neuroscience. We look forward to seeing you there.